Okay, so now we're going to move into some cell signaling that involves G proteins. And so G proteins are going to be important for our glucagon or epinephrine. They're all going to have the secondary messenger cyclic AMP, so this is a secondary messenger. And the cyclic AMP is going to be made, and it's going to activate protein kinase A. We've already discussed protein kinase A, and protein kinase A has regulatory elements of cyclic AMP release. And then you're going to get a signal transduction pathway. So what we want to talk about first is our G protein coupled receptor. So again, we have our G protein coupled receptor, which will bind to some sort of ligand. This can be epinephrine, this would be glucagon. And this ligand receptor bound will recruit a G protein. And this G protein will get activated. Now these receptors are a trimeric G protein, so they have an alpha, a beta, and a gamma. And it's the alpha that gets activated and dissociates from the beta and gamma. And then the alpha is going to go on and activate some downstream enzyme. And then you're going to get some substrate made from this. And so this is an example of a beta androgenic receptor. A beta androgenic receptor, and for example, this one can detect epinephrine, norepinephrine. Oops, just put it. Okay, now G proteins receptors are going to, again, be similar to RAS. We talked about RAS, if you remember. RAS was a G protein. We're going to see RAS pop up again. And RAS with GDP is off. And then RAS with GTP is on. And it's the same with our G protein receptors. With GDP, this is off. With GTP, this is on. And just like RAS, our G protein receptors have the ability to hydrolyze and turn off, but it's an auto turn off, so it's the same concept as we talked about with RAS. Now our G protein receptors have three essential components. They're going to have a plasma membrane receptor with 7 transmembrane alpha helical segments. So basically it looks like a little snake. 3, 4, 5, 6, 7. They're going to have an internal and external side, of course. They always have a G protein that's going to cycle through active, which is GTP bound. So here's active. And inactive, which is going to be GDP bound. And then you're going to have some sort of enzyme or channel that's going to be regulated by the G protein. So here we have our enzyme that's activated by the G protein, and then it's going to transduce some sort of signal. Now the second messenger, here's our second messenger, is going to impact all the downstream targets. So deficiencies of G protein receptors are associated with blindness, diabetes, allergies, cardiovascular defects. About half the drugs on the market are going to target a G protein receptor. These are called beta blockers. So they're very important medically. And the beta blockers are targeting the beta androgenic receptor to treat hypertension, glaucoma, anxiety, migraines, etc. So again, we have our G protein receptor, that 7 membrane alpha helixes. That's shown a little bit better here. The extracellular binding domain is going to be specific for the hormone. So in this case, we're showing epinephrine. So epinephrine is going to bind. The receptor itself has no kinase activity, so that's important. So the receptor so it cannot phosphorylate anything. It initiates all its signal transduction through G proteins. That's why it's called a G protein receptor. And then your different receptors are going to have different specific hormones. So this beta androgenic is a G protein coupled receptor that's in muscle, liver, and adipose tissue. And so some things to keep in mind is this G protein receptors are going to be associated with a trimeric G protein. So this is our G alpha, G beta, and G gamma. And then you're going to have the S. Let me see if I can highlight the S. The S shown here is for stimulatory. So that's going to be a stimulatory G protein. You could also have an I. An I would be for an inhibitory. Okay, and so basically you're going to have a hormone bind, you're then going to have the G protein get activated, so you're going to replace GDP with GTP, and you're going to have your G activated alpha subunit. This is going to activate adenylyl cyclase. Adenylyl cyclase is going to make cyclic AMP, yeah. And then this is going to activate protein kinase A. It can also become a 5' AMP when it's degraded, and so this is going to turn things off. Activation of protein kinase A is going to initiate a response to epinephrine. Now your adenylate cyclase is an integral protein, so this is spanning the membrane. And this sort of integral protein has the active side on the cytoplasmic side. And then again, adenylyl cyclase is what actually makes cyclic AMP. Now, adenylate cyclase, again, forms cyclic AMP, and cyclic AMP is the second messenger for G protein receptors. You have a cyclic AMP phosphodiesterase, and this is what, don't like that color. And this is what's going to turn off this cyclic AMP, so it breaks cyclic AMP down. So basically, you start with ATP, you then are going to generate cyclic AMP through adenylyl cyclase, and then cyclic AMP phosphodiesterase is going to turn cyclic AMP into AMP, and this turns off. So this is on, and then this one is off. Now, cyclic AMP, of course, regulates protein kinase A, as we know, but there are a few things we need to add to the story here. So we already know that cyclic AMP binds to the regulators of protein kinase A, and that releases protein kinase A. Cyclic AMP also has the ability to open sodium channels in some cells, and so it can be involved in ion transport. Protein kinase A is going to be involved in activation of enzymes that are going to mobilize energy, and they do this by phosphorylation. They are a kinase. So protein kinase A is going to phosphorylate phosphorylase kinase and make that active. And then phosphorylase kinase is going to activate glycogen phosphorylase. And then you're going to start to break down glycogen. So this is breaking down glycogen. Now, another thing that protein kinase A can do is that you have the protein kinase A is going to be able to enter the nucleus, and protein kinase A is going to phosphorylate, because it's kinase, a transcription factor, which is the Cre binding protein, or CREB. And CREB is going to interact with genes with Cre response elements. Cre response elements are cyclic AMP response elements, and these are going to turn on genes. So this becomes a transcription factor. And so this is an important pathway because it's going to be stimulated, for example, in response to not only glucagon, but also epinephrine, and it's all to make those enzymes in order to mobilize energy. And so when you need more energy, your genes are going to get turned on by your protein kinase A phosphorylating CREB, which then binds to the Cre sequences in the DNA that turns on whatever gene is downstream of those Cre genes, or Cre sequences. Now, as I mentioned before, our G proteins have their own switch on and off, and so you have your G protein receptor. You get activation of your G protein alpha by the switch from GDP to GTP, and then you're going to have hydrolysis, and you're going to lose a phosphate, and your receptor is going to reset and wait for another stimulator to bind to it so it can go through the process again. Now this is an example of signal transduction and amplification. So up here you have one molecule of epinephrine, and it's going to activate that G protein. And so you're going to get 10 molecules of your G protein activated, your adenyl cyclase. And then after you have adenyl cyclase, you're going to make cyclic AMP, so you might make 200 molecules of cyclic AMP. And then you're going to activate protein kinase A. You're going to make about 100 molecules of that. You're going to activate phosphorylase B kinase, 1,000 molecules of that. Glycogen phosphorylase A for active, 10,000. And you're going to release glucose. And so you're going to have 100,000 glucose molecules for that. And so this is basically the signal transduction for that very fast on enzyme control mediated by epinephrine signaling. Now there are other G protein receptors. We have hormones that activate different receptors that have different signal transduction pathways. And some of those can lead to phospholipase C activation and phosphatidylinositol activation. And we have an example of this is the alpha-1 androgenic receptor, which causes contraction. So in this case, you still have your hormones. Your hormones are still binding to the receptor. So you have your you have two intracellular second messengers in this case. So in the other one, we had cyclic AMP. But here we're going to activate two. We're going to activate your two receptors that are going to be generated from phosphatidylinositol, which is PIP2. So again, we have our hormone that's going to bind. You have your alpha unit that becomes activated. In this case, is phospholipase C that gets activated. Phospholipase C is basically going to cleave phosphatidylinositol 2, which is phosphatidylinositol 4,5-bisphosphate. And then you're going to generate your diacylglycerol here, and you're going to generate IP3, inositol phosphate. Diacylglycerol is going to be one of your secondary messengers, and IP3 is going to be another of your secondary messengers. And so IP3 is going to be able to open your calcium channels, and diacylglycerol is going to be important in activating protein kinase C. Protein kinase C activation by diacylglycerol and calcium, which is caused by inositol phosphate 3, are going to lead to more cellular responses. So you're going to activate different kinases, you're going to have phosphorylation, etc., downstream of all of that. But it all starts with the binding of the hormone receptor, the binding of the G-alpha, this time to phospholipase C, the splitting of phosphatidylcholine and inositol into IP3 and diacylglycerol, or DAG, which sometimes is DAG. And then those two secondary messengers are going to activate protein kinase C eventually, and then you're going to get more activation in the cell. Now, our signals from G proteins can be either stimulatory or inhibitory. So we have a G-alpha stimulatory, and so examples of hormones that use that are going to be glucagon, epinephrine. Cholera toxin is going to modify the G-alpha stimulatory and keep it active. And that's one way cholera toxin causes its effects, because basically if you have in your gut your G-alpha proteins activated, you're going to open ion channels, and sodium is going to be released, and water follows sodium, so you get a massive release of water into the gut, and then that leads to the symptoms of cholera. We have G-alpha inhibitory O, which is going to inhibit adenyl cyclase. Again, epinephrine neurotransmitters can use this. This is a target of pertussis toxin, and pertussis toxin is going to inactivate this G protein. There's also the G-alpha Q11, which activates phospholipase C beta version. And so epinephrine, acetylcholine, and histamine can all activate these receptors.